Hello friends, in the last lecture we have studied the mechanism of enzyme action. In that lecture we have studied how the enzyme substrate complex get formed and the energy state in the enzyme catalysis we have studied. We have studied the Michaelis-Menten constant and many more things. So the mechanism of enzyme action can be easily explained by two hypotheses, and that are the log-key hypothesis and induced fit hypothesis. So you are welcome in this lecture and you are studying the mechanism of enzyme action with Yuvraj. So basic reaction of the enzyme substrate is as it is takes place in two steps. In the first step, enzyme combine with substrate to form ES complex, and in a second step, the enzyme substrate complex form the product. That product is get formed and enzyme liberated from the reaction. The action of mechanism of enzyme can be explained with the help of two hypotheses, log-key hypothesis and induced fit hypothesis. The log-key hypothesis was proposed by the scientist Emil Fischer in 1890. The explanation of enzyme action on the substrate was given by the scientist Paul Fields and D.D. Woods. As per the log-key hypothesis, every lock has specific key. That key can open the lock or close the lock. Like that way, the every substrate are specific to the enzymes. The enzymes are specific to the substrate. So this is called as enzyme specificity. The key is specific to the lock. So key substrate get fitted into the enzyme. Enzyme substrate complex get formed and it is based on the specificity of key to the lock. See here, the figure said that there are three substrates, X, Y and Z. The yellow colored enzyme is also there. So when we look the active sites of the enzymes, the three active sites are there, A, B, C. So the substrate who having the exact complementary active sites, the substrate who having the exact complementary structure to the active sites can only bind. So look here, the X will not be bind to the enzyme because it has only two furrows. Z cannot also bind to the enzyme because it has only one reach, and Y can be exactly fitted into the active sites of enzyme A, B, C. Therefore, Y is fitted into the enzyme and form the ES complex. So, substrate Y is the key and enzyme is a lock. So it is fitted exact complementary to the enzyme. So this is the lock key hypothesis. The active sites are rigid here, they are permanent, they do not change their structure, conformation, and that's why this model is called as lock key model. So every enzyme has active sites. These are formed by the amino acid and their residues arrangement. These active sites are rigid, they are not flexible, they not change their conformation structure, and they are strictly complementary to the substrate. They are meant for attachment of substrate to the enzymes, and finally the product get formed and enzyme get released. So the enzyme has peculiar active sites, like lock has a specific key, and that's why it is called lock key hypothesis. The second hypothesis is induced fit hypothesis. This hypothesis was proposed by the scientist D. E. Coastland in 1966. The substrate induces some conformational changes in the enzyme structure, and enzyme substrate complex get formed, that is called as a induced fit hypothesis. म्हणजेच काय तर एन्झाईमला इथं ॲक्टिव्ह साईट असत नाहीत, सबस्ट्रेट त्यांना जशा पाहिजे तशा ॲक्टिव्ह साईट्स एन्झाईममध्ये तयार करतो आणि तो एन्झाईम बरोबर बाइंड होतो आणि एन्झाईम सबस्ट्रेट कॉम्प्लेक्स तयार होतो. इंड्यूस करणं म्हणजे तयार करणं, एखाद्याला प्रवृत्त करणं, ॲक्टिव्ह साईट्स तयार करण्यासाठी प्रवृत्त करणं. सो सबस्ट्रेट गेट इंड्युसेस दी ॲक्टिव्ह साईट्स इन द enzyme. Hypothesis was proposed by the scientist D. E. Coastland. So see here the substrate, green colored substrate, you are looking here, an yellow colored enzyme. This green colored substrate induces the active sites in the enzyme, and enzyme substrate complex get formed. So main features of induced fit hypothesis are the enzymes having active sites, but that active sites are not rigid, they are flexible. They change their structure conformation as per the nature of substrate. The enzyme substrate are not strictly complementary to the each other. The active sites are not rigid, they are flexible one. This is the same as the hand gloves. When we insert hand into the gloves occupy the space. Gloves, the rubber gloves increases its size as per our hand, and it get correctly fit with our hand. That's why the model is called as a hand gloves model. So this theory of enzyme substrate interaction has two advantages compared to the lock-key hypothesis. First one, it explains how enzymes may exhibit broad specificity. Example, lipase can bind to the variety of lipids. And second, it explains how catalysis may occur. The conformational changes stresses bonds in the substrate, increases the reactivity. So how the catalysis occur, that can be studied with the help of induced fit hypothesis. So look here, the difference between the log-K hypothesis and induced fit theory. In a log-K hypothesis, the active sites is like a lock to which the substrate fit like a key. The enzyme active sites and substrate should fit like a lock and key to the initiate the reaction ES complex. Induced fit hypothesis, this is also called a hand in a glow model. So hand glow model, the binding of a substrate induces the conformational changes in the active sites, and both adjust their shapes to provide optimal fit. So this is all regarding the log K hypothesis and induced fit hypothesis. Thank you very much.